Okay, so let's say we've got a neuron here, right? That's going to release a neurotransmitter. It's going to, there's going to be an action potential arriving, depolarization, opening of the voltage-gated calcium channels, interaction with the docking complex, fusion of the vesicles, release of neurotransmitter that's going to traverse the cleft and then bind to an ionotropic or inotropic receptors that are located postsynaptically at this axodendritic synapse, right? And let's say this is going to be an excitatory, impact on this next cell. It's going to actually push it in, towards threshold, so it's going to depolarize it. Well, if you have a neurotransmitter, let's say it's glutamate, because glutamate is our primary excitatory neurotransmitter in the brain. But it's excitatory because it binds to, in this case, postsynaptic, ionotropic receptors, also known, remember, as ligand-gated channels. And the consequence of that binding is the opening within that ionotropic receptor, right, of a channel that lets sodium pass through. So, glutamate is excitatory because it binds to a postsynaptic glutamate, receptor, an ionotropic receptor that opens a channel that lets sodium enter, because sodium is going to, of course, come in. It's driven by the concentration gradient and the electrical force, and that entry of that, positive sodium is going to depolarize the, postsynaptic cell. So it's going to have an excitatory impact because of the ligand-gated channel, the glutamate receptor, the ionotropic receptor that it binds to. And yet, one, glutamate releasing neuron, releasing glutamate at one axodendritic synapse here, and binding to a couple of, postsynaptic ionotropic receptors, opening, small sodium currents. While it will depolarize a little, it's not going to be sufficient, to set off, an actual action potential to depolarize to minus 55 here at the axon hillock. So what's happening is, the way it's described, we say that when, there's release of, let's say, glutamate, it binds to these ionotropic receptors, which open sodium channels. Sodium comes in, and there is a small, depolarization. It's called an excitatory postsynaptic potential, or an EPSP. There's a slight movement, from minus 65 at the resting potential at this particular spatial location where, the receptors were stimulated, in the depolarizing direction. And you can also have the opposite. So imagine there's another neuron here. This is a different one here. This is, let's say, an inhibitory neuron. And it's going to release a different neurotransmitter. Our primary inhibitory neurotransmitter in the brain is one called GABA. GABA is going to, traverse the synapse, bind to its own postsynaptic ionotropic receptors or ligand-gated channels. But unlike the glutamate receptor, which opened a sodium hole, the GABA receptor is going to open a channel that lets chloride go through. And remember, chloride's in high concentration outside the neuron, so it'll be driven in by the concentration gradient force. It's also opposed, of course, its entry by the fact that it's negatively charged on the inside, and it's a negatively charged ion. But at the resting potential, anyway, the concentration gradient force is stronger. It wins. And the entry of some negative chloride ions into that postsynaptic cell will result in a small hyperpolarization, right? So this, again, is going to be a inhibitory postsynaptic potential. So this is an IPSP. It's not enough, just one of these, or a few of these, to generate full inhibition, but it leads towards more inhibition. So imagine you've got your typical neuron here, right, with up to 10,000 distinct synaptic connections and all these dendrites. Well, some of them are releasing neurotransmitters that are binding to ionotropic receptors that will open sodium or calcium, depolarizing currents, small EPSPs, while others are, releasing neurotransmitters that are binding to ionotropic receptors postsynaptically, that open chloride or potassium currents, right? So they'll be, small IPSPs. So this cell is going to do a lot of summation here, and we're going to see that summation is going to be occurring here, at the axon hillock. if you can, get to minus 55, get to the threshold right here, you can open all those voltage-gated sodium and potassium channels and initiate an action potential that'll propagate, towards the end of the cell. But it's made up of all these small EPSPs and IPSPs, that are spatially and temporally distributed all around